Hi everyone, my name is Madeline Blake and I'm an incoming junior at the University of Minnesota Twin Cities with a triple major in genetics, cell biology and development, neuroscience, and cellular and organismal physiology. This summer I've had the opportunity to work in the Prince Lab investigating the impact of therapeutic ketosis on pulmonary arterial hypertension. Pulmonary arterial hypertension is a rare but lethal condition characterized by elevated pressures in the pulmonary vasculature, which ultimately leads to right ventricular failure and death. In this pathophysiological transition to right ventricular failure, several metabolic impairments occur, of which includes depressed fatty acid oxidation, which leads to a decrease in energy production. This decrease in energy production exacerbates existing impaired RV contractility, and therefore interventions that are aimed at improving cardiac metabolism and RV dysfunction hold a lot of therapeutic potential, which is why we decided to look at ketones. So ketones are an alternative fuel source produced by the breakdown of fatty acids in the liver, and with these ketones, we think that that may be a very promising non-pharmaceutical intervention for pulmonary arterial hypertension. So our experiment involved the administration of a ketogenic diet to rats treated with pulmonary arterial hypertension to see its effect on right ventricular function and hypertrophy. We found that this ketogenic chow actually went ahead and improved RV function while not changing disease severity or hypertrophy, which establishes therapeutic ketosis as a very promising non-pharmaceutical intervention for pulmonary arterial hypertension and other disorders involving heart failure.
My name is Suprata Chavli, and this summer I've been working in the OGA lab, which is working on creating and studying engineered heart tissues, or EHTs. These EHTs are generated in a rectangular PDMS mold with two rectangular posts on either end. While this design is functional, there are a couple limitations. The first being that it's too big, and the second being that the EHTs cannot be removed from the PDMS, which makes viewing them through a microscope a little difficult. So this summer we designed a new mold that is made with 3D printed spacers casted with agarose gel, and for the posts we're using steel needles bent into an L shape and attached to a 3D printed lid that goes over the well. It is expected that this design will maintain the functionality of the old design while fixing a couple of the limitations that it had.
Hi, my name is Solana. I'm a rising senior at Macalester College. This summer I'm working in the Mary Gary lab using a method called direct cardiac reprogramming to grow blood vessels. So blood vessels are essential for the transportation of oxygen and nutrients throughout the body, but following heart attacks and ischemic injuries, they can become damaged or destroyed. And after a heart attack, something called a fibrotic scar forms, which is basically this growth of fibroblast cells. Initially, this is kind of an injury response mechanism, but the growth gets so bad that it obstructs blood flow and actually prevents healthy heart function. So what we're trying to do is take these fibroblasts that are growing and treat them with different chemicals, different compounds, and activate genes of interest so that they actually reprogram into endothelial cells, which are the cells that make up the vasculature. This is a super exciting therapeutic option and alternative because the current standard of care for heart failure is just a heart transplant, which a lot of patients need, but very few end up actually receiving.
Hi, my name is Maddie Kelly and I worked in the LHI lab this summer. Heart failure is the condition in which the heartbeat becomes weak or abnormal, preventing blood from circulating throughout the body. It is one of the leading causes of death in the world despite various types of treatment available. Thus, the call for new research is extremely important. My lab focuses on the pathological mechanisms behind heart failure in order to develop new drug therapies specifically targeting mitochondria function. Even though mitochondrial dysfunction may be one of the causes of heart failure, there is no available clinical drug or treatments that specifically target the mitochondria. Mitochondria are the powerhouses of the cell, generating energy through processes that are triggered by calcium accumulation. Too much calcium accumulation, however, may cause oxidative stress, which is the buildup of reactive oxygen species leading to cell damage. Mitochondria-mediated oxidation appears to be one of the causes of heart failure, primarily because of the MCU, or mitochondria calcium uniport channel. Mitochondrial calcium accumulation depends on one channel, the MCU, and the amount of calcium that goes through the MCU is regulated by multiple cell signaling processes, including phosphorylation, which is a type of protein modification. Phosphorylation of the MCU causes more MC units to be inserted into the mitochondrial membrane, increasing the amount of calcium accumulation. This summer, my project specifically focused on why the MCU is phosphorylated and how to detect this level of modification in human heart failure. My research is very important because this protein biomarker may be helpful for understanding the severity of human heart failure and can also be useful in developing new drug therapies that prevent cell damage in patients.
Hi, I'm Lydia, and I've been working in the Dan Gary lab this summer. My project focuses on growing blood vessels through the direct reprogramming of fibroblast cells to an endothelial lineage. Endothelial cells make up the inner lining of blood vessels, which are essential for transporting nutrients and oxygen throughout the body. Being able to reprogram fibroblasts to endothelial cells is exciting because doctors could then control blood vessel growth, which opens the door for new therapies. For example, treating cardiac fibrosis after a heart injury. So I'm looking at overexpressing the pioneer factor, ETV2, which has been found to affect and play an important role in the development of endothelial cells. So I'm looking at overexpressing this gene through the addition of doxycycline, an antibiotic. Past studies have found that doxycycline overexpresses ETV2 in mouse embryonic fibroblasts, causing the direct reprogramming of fibroblasts to endothelial cells. And so I'm looking at adding different transcription factors and drug compounds along with doxycycline and observing their effect on the reprogramming process. I'm looking at whether or not they continue to further amplify ETV2 gene expression with the hope of making the reprogramming process more efficient and effective.
Hi, my name is Sydney, and this past summer, I have been working in Dr. Kaiba's lab on investigating the effect of estradiol, the main form of estrogen, on proliferation and differentiation of human myoblasts. Estrogen is an essential sex hormone, and loss of estrogen in postmenopausal women leads to a condition called sarcopenia, which is characterized by declines in muscle strength, mass, and regenerative potential. Recent studies have established the beneficial effect of estrogen on satellite cell growth in mice, but much less is known in humans. And so in my studies, I have found that estradiol caused human myoblasts to proliferate to a greater extent than the vehicle alone. And greater concentrations of estradiol led to a higher growth rate. Estradiol had little to no effect on the differentiation, but nevertheless, these results suggest that estrogen promotes proliferation of human myoblasts and supports a model in which impaired muscle regeneration in postmenopausal women is due directly to the lack of estrogen signaling in skeletal myogenic progenitor cells.
Hi, my name's Inbaraja Shankar, and this summer I worked in the Von Berlo lab, studying the role of transmembrane protein 100, or TMEM 100, in regulating cardiac fibrosis. Fibrosis is the excess deposition of extracellular matrix proteins, mainly by activated fibroblasts, forming scar tissue. This process aids in the healing of the heart after injury, but could be detrimental to overall function in the long term by reducing cardiomyocyte relaxation and impairing electric conductivity. Moreover, fibrosis is present in many cardiovascular diseases that result in patient injury or death, so it's important to find ways to reduce it. Although we found that TMEM 100 expression remained constant after TAC injury, by inhibiting it through siRNAs, we noticed a decrease in fibroblast differentiation and a reduction in overall fibroblast counts in cultured cells. Furthermore, inhibition of TMEM 100 resulted in an increase of fibroblast calcium levels, a major cause of cell apoptosis. In cardiac fibroblasts, through the regulation of differentiation and the modulation of calcium levels leading to cell death, our results show that TMEM 100 plays an important role in regulating cardiac fibrosis and in potential treatment strategies moving forward.
Hi, my name is Iris, and I'm a rising junior at the U of M. This summer, I've been doing research in the Perlingero lab on the effects of Duchenne muscular dystrophy on striated muscle. This type of muscular dystrophy is caused by an absence in a protein called dystrophin, which causes patients to first experience muscle atrophy in their skeletal muscle and then later on in their cardiac muscle. In my project, we differentiated heart and skeletal muscle from unaffected human stem cells and stem cells with Duchenne muscular dystrophy, and then subjected them to contractile stress via electrical pacing and chemical stimulation. After stimulation, we measured the creatine kinase level of the cells, which is a diagnostic measure based on levels of muscle damage. We hypothesized that the cells with Duchenne muscular dystrophy will have higher levels of creatine kinase than their unaffected controls, and we hope these results will help pave the way to understanding the differences between the responses in heart and skeletal muscle.
Hi, I'm Derek Williams and I'm here in the Townsend lab. Duchenne and limb-girdle muscular dystrophies are progressive diseases of muscle degradation thought to cause little tiny rips in the muscle cells' membrane, effectively killing them. My project's approach to that issue is that we could use a special agent called Ploxmer 188 to act as a little molecular band-aid to help repair and protect these cardiac muscle cells. To test this, we injected P188 in both a limb-girdle and Duchenne mouse model, and upon analysis of their hearts we found that only male Duchenne mice were significantly protected from damage, suggesting that using P188 to repair rips is a little bit more complicated than we thought, but perhaps more interesting is the apparent difference between Duchenne and limb-girdle, previously thought to be very similar diseases.